Professor William Mobley will be chairing the meeting for the day, and if we could introduce him. Thank you. And as I say, we're running to a tight schedule, so try to be here.
Let me do a little bit of housekeeping before getting started. You will know from the emails and from the correspondence with Peter that we've planned a slightly different conference than is usual. We'd like people during their presentations to refer, essentially to give us a status report on their fields. What is their field and what are the strengths and weaknesses of work that's going on so far, and what are the gaps, what are the things that we must do that we haven't done, what resources will be needed to fill gaps, how can we as a community assist one another in doing that, and then what expectations are there for enhancing the lives of people with Down syndrome if the gaps are filled. So this, for me, is a very important list, and this meeting's different, and I thank Peter and his foundation for making this possible, because we don't usually go at it quite this way, but I think if we focus on these questions, we will have a lot more specific kinds of ideas that come forward and that we can act upon. Now, you'll know that later in the day, there are roundtable discussions, and I wanted to just remind the speakers, but announce more generally, there'll be 3 pre-roundtable meetings of about 40 minutes. Roundtable Group 1 will focus on basic science and biomarkers, and the speakers that will be involved are Potter, Obeid, and Tycho. But of course, anyone in the audience interested in the basic science of Down syndrome and being part of that group, please join that. I don't know where they'll meet, but I presume that one might meet here and one there, and perhaps one in the back. We'll figure that out later. The second roundtable group focuses on clinical issues and clinical research tools, and Tournay, Simmons, Ercole, Sumich, and London will be part of that group, but please join it if you're interested. And finally, Roundtable Group 3 is really focused on translation, if you will, the bringing of treatments to people, and that's Lister, Dearson, Adak, Smith, and Revsum. So these groups will meet from 3:20 to 4 o'clock. Then we'll have a joint session to report out from the roundtable discussions from 4 to 4:40, and then the last 20 minutes or so, we'd like to have a summing up of the meeting and proposals for going forward. And I'll have a little bit more to say about one proposal I would make for going forward at that time. Let me begin then my slides. I'll show from 2 different decks. So we are interested at the cellular and molecular, but also the behavioral and sort of brain level at cognitive circuit failures. What I think we can say so far is that we've defined a number of relevant phenotypes in mouse models. We have some success in defining genes and mechanisms, which is our search. Our search really is very much on finding the genes and mechanisms involved. And we've had our first engagements with industry. I think of that as a success. The weaknesses is that we've really only scratched the surface of many of these phenotypes. I suspect that we've really able to identify 120 different phenotypes in the mouse brain that we think are relevant to the human brain and of the person with Down syndrome. But really, for most of these, we're still looking for genes and mechanisms, and as you know, there are no treatments yet tested in people. So I'll come back to this, but I think there are many gaps to fill in essentially defining the genes and mechanisms that we need to understand to make progress. So I'll come back to these gap slides later. Let me just mention very briefly our work in summary form. I work at the University of California, San Diego in the newly announced Center for Down Syndrome Research and Treatment. Our plan is quite simple. We have a boss, and one of our bosses is this little boy, this little pirate, and he's as cute as he could be. We are very interested in beginning with the problems people have, children and adults, to work then toward discovering genes and mechanisms in mouse models, to proposing treatments and to bringing those treatments to the clinic. As mentioned, a very difficult foray, but one that we think is already yielding fruit. So the hypothesis is simply that an extra gene or genes are responsible for abnormal synaptic function, that brings on cognitive problems. And the research strategy then simply is to define those phenotypes as carefully as we can at every level, from the molecular to the behavioral, to search for the genes and mechanisms, and then to, using that information, to attempt to create new treatments. We use mouse models, and here's a diagram of where we stand today.
Can you put it on the show?
Oh, I'm sorry, I will. Yeah. So here's human 21, and here's mouse 16, and mouse 17 and mouse 10, those portions of those genes that carry mouse homologs of the human, the long arm of human 21. And here are the names of mice, excuse me, that exist. So there's Eugene used DP16, DP17, and DP10, and you may know that there is a mouse that in fact incorporates all 3 chromosomal fragments. That mouse, by the way, does exist. It's available to you through Jackson Laboratories right now, as are each of these 3 smaller fragments. Much of our work, and in the field, much of the work has been done on the TS65DN mouse, and the data that I'll show you is largely from that mouse model. Mouse models have been critical for progress in the work that we've done. So there are 2 themes. The first theme looks at an imbalance in excitation and inhibition, and we're exploring the mechanism, and we think we have a gene segment that's terribly important for that. And we can say that drugs that reduce GABAergic transmission enhance cognition. And there's a second theme, one that's much more relevant to the adult with Down syndrome, that looks at the disassembly, if you will, of circuits later in life. And there a gene has been identified, only one of probably several, but one very influential one. And I'll tell you a little bit about the mechanism that we have looked at there. So on the first theme, what we can say is that inhibition is excessive. We know this to be the truth in the hippocampus. We believe it strongly to be the case in cortex as well, though there's much less data for cortex. In a nutshell, there's increased activation in the mouse model, in the slice, if you will, of both GABA A and GABA B receptors. Both, when activated, reduce synaptic plasticity, and antagonists of each of these different receptors can restore LTP and memory. What you've heard about mostly is the GABA A story, but I think you'll be hearing very soon much more about the GABA B story. And we see these as complementary. We see that the circuit biology in the mouse is heavily influenced by an imbalance of excitation and inhibition that favors inhibition. So we believe strongly that this imbalance is quite critical for compromising cognition. Excuse me. So targets to modulate or normalize inhibition include GABA A receptors, and there, selectively reducing activity of GABA A receptors makes sense. There are compounds that do exist. We've not just been in active dialogue with Roche, but in fact are testing some of their compounds in our mice. And they have, as you know, announced a clinical trial. And I consider this one of the most amazing and helpful and important developments in all of Down syndrome. I think 5 years ago, to think that there would be a clinical trial in people with Down syndrome was almost unbelievable. But it is happening, and happening with a terrific company, with a terrific plan. The second approach would be to selectively reduce the activity of GABA B receptors. There are many safe compounds for this, but none of them are in development at this point. And then something that we would hope for, that we think may be very helpful, is the ability to selectively reduce the activity of GERK2 channels that are encoded by the KCNJ6 gene. A very attractive target for us because it addresses the fundamental pathology at the genetics level, but there are no selective compounds as yet, and there are no industry partners. The second mechanism is the inadequate maintenance of circuits, and you may know we have been very interested in studying the biology of trophic signaling, and in essence to understand how circuits are maintained through the signaling of trophic factors that bind to their receptors on axonal terminals, are enveloped in endosomes that carry signaling proteins that are moved back to cell bodies. I think we can say with some confidence that this is quite abnormal in people with Down syndrome, in people with Alzheimer's disease, and in our mice models, and it's registered in people's brains through the production of very large endosomes that we presume move poorly. Here we're looking on the top two chymographs. What we're looking at here is distance, this is time here, and this is distance moved. And so what one can see over time is that the endosomes that carry trophic factors, labeled here through quantum dots, move rather smoothly with some pauses and very little anterograde movement. So here we're moving from the axon terminal to the cell body. In the bottom panel is the TS65DN chymograph, and what you can see here is there's much more anterograde movement, there's much more pausing, and some pauses are quite long. The net effect of this, we believe, are cells that simply don't see the trophic signals in their terminals and that thereby become atrophic, dysfunctional, and ultimately disappear. What we can say about this is that we know, in fact, that these endosomes in the TS65DN mouse are too large, and I don't show you a figure here that plots all the numbers out, but in fact these endosomes, as you can see here, are quite a bit larger and really fill both cell bodies and processes. We know now that this is the case in the TS65DN mice, and we've implicated APP as a major player. In particular, we've implicated the beta stub of APP, this processed product as a result of beta secretase. We've implicated this as an important player, and I can be happy to show you data later, we know in fact that overexpressing either full-length APP or beta stubs increases the activity of this GTPase called RAB5, and that increase results in a marked increase in the size of endosomes, and we believe a marked decrease in their transport. In essence, what's happened is that you've isolated the terminal of these mini neurons in Down syndrome, the terminal from the cell body, and there are two consequences to that. The first consequence is that the cell body becomes atrophic. It simply doesn't see its target. It's no longer in intimate contact with its target. The second problem is that there's a massive increase in signaling in the terminal. And I'll just show you here the results of some studies just completed with Steve Smith at Stanford with my student Rachel Noshaney, using an elegant technique called array tomography, in which one can look at the light level at literally hundreds of thousands of synapses. And what she's shown here is that if you classify synapses on the basis of their immunostaining properties, and here she's looked at 100,000 synapses, one can actually size them, characterize them, and count them. And what she's seen is a dramatic increase in both the number and the activity in synapses, GABAergic synapses of tract B, the receptor for BDNF. So what we have is signaling gone wild in these synapses, selectively GABAergic synapses in the mouse model of Down syndrome. So returning to our strategy, the cognitive phenotype here is age-related normal learning and memory that degenerates abnormal learning. We know that APP gene dose plays a role, and we'd love now to do one of several things: decrease the level of this protein, or modify its processing, or restore connections. And what I can say is there's real success here on the way. We've been able to change the processing of APP, at least in culture. We've had recent success working with AC Immune through an antibody to reduce A-beta levels. And we have previously published a paper in which, by restoring levels of norepinephrine, we can enhance learning and memory. Let me mention just the first piece very briefly. What we see using something called a gamma secretase modulator is the ability to enhance the activity of gamma secretase, thus processing stubs into A-beta and processing A-beta into small A-beta fragments. So rather than an inhibitor of gamma secretase, which we would argue would make cells sicker, and in fact in clinical trials makes people less cognitively functional, a different approach, an approach that likely involves allosteric modulation, increased activity of presenilin and a gamma secretase complex to produce smaller, less toxic fragments of A-beta and to clear the very markedly increased levels of beta stubs that one sees in the Down syndrome brain. So we believe that there are now at least 6 rational targets to pursue. 3 for each. An approach that would focus here on GABA, and an approach here that would focus on normalizing endocytic pathways. Here are the folks that I work with: Pavel Belachenko, Alexander Klishevnikov, Ahmad Salehi, Mike Maloney, Qingbiao Wu, and newer members of our group, April Weissmiller, Steve Wagner, and Rachel Noshani, and here's the support we receive. So I don't know where I'm in my time, but I'm going to stop here and we'll take questions. Are we okay, Mara, on time? How much do we have?
5 minutes.
Do we have 5? Well, if it's less than 5, Mara, you'll tell me. I'd be happy to take questions. Yes, ma'am.
You mentioned it was GABA B receptor.
Yes.
If you are maybe targeting GABA B1, and that can you see the level of GABA B1 in early stage of Down syndrome?
Yeah.
Aging effect?
Yeah, so what I can say is that in the mouse model, there's no change in either the concentration of R1 or R2. So it is not that these receptors are overexpressed. Instead, we think they're more effective, and in fact, we have evidence to say that they're more effective. You get bigger currents with baclofen in the TS65 than in the 2N, but we think that's really simply a consequence of the increased expression of the GERK2 channel, which really lets more potassium out and more effectively depolarizes these cells. Let me very briefly say that there are gaps to fill. To summarize it very briefly, we really need to learn much more about changes in synaptic structure and function. We need to learn much more about inhibitory mechanisms. We need to learn more about reductions in neurogenesis, and I think Mara may speak to this. We need to know much more about how trafficking fails, and we want treatments that actually work. And part of this must be a robust engagement with industry. So what are the resources? Mouse models that are readily available, including a list or a library of mouse models in which selective genes have been deleted or triplicated. A network of laboratories that collaborates effectively. A human clinical and tissue bank, as was referred to by Henri, and a clinical trials network, very helpful. A therapeutic development plan in which there's an ongoing engagement with industry, building with industry a capacity to move discoveries to drugs by having this clinical trials network, and then a consortium of investigators whose universities agree to share intellectual property and move things forward. Of course, there's need for funding. It'd be lovely to have a consortium approach to encourage novel, cross-disciplinary initiatives, to increase visibility for this work, and really to politic for increased funding for Down syndrome, which is very much needed. So I would like to see us work together to define priorities and collaborate. And also, I'd like to see us achieve work that would enhance the lives of people with Down syndrome and their families. So I think we have a dream together that life can be better for these folks, that they can live more independently, richer lives. We love them. They're very valuable human beings, and yet we know there are things we really must do to enhance their life happiness, the things they do in their life. And hopefully, one day be able to say to parents of a child with Down syndrome, guess what? Research has progressed beautifully, and you don't ever have to worry your child will never have Alzheimer's disease. It just won't happen. So that's the hope. And maybe one last question, and I should stop. Yes, sir.
My name's Chris Brennan. I'm a parent of...
Well, what I would say, let's take the easy case. The easy case is the late onset change in brain pathology and dementia. The richest by far, if you want to use that word, genetic source of Alzheimer's disease is Down syndrome. We already know enough about mechanisms, those mechanisms that lead to endocytic pathway failure, that are likely to be highly common in Alzheimer's disease and Down syndrome. In fact, the compound that we're working on, a version of that compound, has already effectively reduced amyloid levels in a mouse model of Alzheimer's disease. So I think at the very simple level, Down syndrome is the perfect conduit to understanding pathogenesis and treatment of Alzheimer's disease. You may know that currently in the US we're spending $180 million in care. By the way, less than $1 in 1,000 is spent on research, but we spend $180 billion on care of people with Alzheimer's disease, and by 2040 we'll spend more than $1 trillion in the US on their care. In 2050, if you count all the people with Alzheimer's disease and 2 caregivers, the population will equal the population of Northern Europe. This is a very important oncoming epidemic. And my view is that we can study it from day 1 of life in people with Down syndrome and make a huge contribution. It's not insignificant that there are that even the treating children would be significant. Were there an effective anti-GABA treatment scheme, in the US alone, you would be treating 400,000 people. That's not so bad. That's almost the size of the MS population, and lots of money spent on treating MS. So I would argue that the number of patients that we have to care for and the plausibility and the rationale of the schemes we're developing already justify a major investment by industry. And maybe we can get your friends at Pfizer to even get more excited.